All right, good afternoon, everyone. I'm super happy to introduce Doc Edge, our speaker for today. Doc is an assistant professor at the University of Southern California. He's been there for 3 or 4 years now. Before then, he was at the University of California, Davis, working with Graham Coop, doing a lot of beautiful modeling of population genetics, and especially how it applies to humans. Before then, he was at Stanford University, where he worked with Noah Rosenberg, doing even more population genetics models. Seems like he's boring at that, but he's not. In the middle of that, he decided somehow like that he had the time to not only have a family, but also write a statistics from scratch textbook that is has been very well received. I don't know how a grad student / postdoc writes a textbook, but he did that. He does an amazing work at communicating science and making genetics important to humanity, to society overall. He is capable of writing commentaries on movies like the 25th anniversary of Gattaca, or is capable of and has the energy to respond to genetic racism online, and has an immense amount of patience for that really, really thankless, awful task. And is a wonderful person to talk to all around. So he's going to tell us about his quantitative genetics, or rather complex trait genetics today, and I'm going to give him the floor. Thanks very much.
So thanks, Raphael. It's very kind. We're going to put the communication skills to the test. It may not live up to that introduction. We'll see. So excuse me here. As Raphael said, my background's mostly in population genetics, evolutionary genetics, and so I wanted to start by just the clicker's working. Maybe not. I might have to... Yeah, let's see here. You're alive. Okay, there we go. Yeah. So I want to just put up for a second. This is my group, and these are the things that I do that I'm not going to talk about today. But these are people who do all the actual the work that I'm going to talk about, and I'll say more about them as I go along. But what I want to talk about today is trees, and this kind of tree, not the kind of tree that actually grows from the ground, but the kind of tree that represents information in a hierarchical structure. And you know, my training is in evolutionary biology, and evolutionary biologists, we love trees. We've been drawing them since before we were really evolutionary biologists, because they communicate this notion of common descent that's so important for understanding how evolution works. And as an evolutionary geneticist as well, we really love trees, not just at this sort of phylogenetic scale, at this long time scale kind of tree, but short-term timescale trees also structure the way genetic variation is going to show up on haplotypes. So if you imagine I have like this sort of musical clef looking notation is a set of haplotypes, and the little dots instead of being notes are you know, mutations on these haplotypes. They're structured in a particular way. It's certainly not you know, independence among the different sites. There's a lot of correlations, and the reason that those correlations exist in the way they do is because these mutations arise from you know, haplotypes that were inherited by multiple times by individuals in the sample, or individual haplotypes in the sample. And so, like, for example, you know, we see that E, F, and G down there are all inherited, you know, all descended from the same common ancestor. You could say they coalesced very recently back in time, and so they tend to share a lot of their mutations, in this case, all of them. And you can see that kind of structure existing throughout the tree. And this is what we often call a coalescent model in population genetics, this sort of haplotype inheritance model. And it's really important for modeling in general in population genetics, and also just a great way to think about how genetic variation arises. And of course, if we're thinking about a whole organism, a whole sexually reproducing organism, we can't think of just one tree as describing the whole genome, because recombination events happen. And so recombination breaks up that pattern, and it does things like, oh, I did the wrong thing, this button. It does things like this, where you can think of a recombination event as like a subtree prune and regraft operation. So you snip off this little tip and it coalesces onto the tree somewhere else. So like in this case, it goes back up here toward the root and it changes the topology of the tree from one place to another. So there's a lot of nodes are the same between these two. It's just the ones that are specifically affected by that regraft operation that change. And so you get this sequence of correlated trees along the genome represented like this. And this sequence is sometimes called the ARG, or the ancestral recombination graph. I'm gonna ignore the distinction between the ARG and tree sequences for the real nerds in the room, and just think of them as being the same thing. So the ancestral recombination graph encodes all of the recombination events and all of the common ancestry events that affect genetic variation in the current sample. So in some sense, it's kind of like a holy grail object in population genetics. If we knew the true ARG, and we don't, except in really unusual circumstances in in crosses that we understand really well and things like that. We generally don't know it in a natural population, but if we did, we would sort of know everything. For people my age, you might think of like Highlander, where he knows everything at the end of the movie, and that's the situation we're in. Us population geneticists, we wax a little bit religious about the ARG. This is Adam Seipel saying, "I believe in one ARG, creator of heaven and earth, of all things visible and invisible," which of course is a reference to the Nicene Creed. Tongue firmly in cheek, but it kind of gets across the notion that this is a really important theoretical object among people who like to think about population genetics in particular. Now that's all well and good, but we don't know this sequence, right? If I just grab some individuals from the population, I don't know when they co-inherited their pieces of their genomes from each other, or what the recombination modes were, and so forth. And it turns out that inferring that is very, very hard, doing it from genetic data. But it has been a huge focus of effort over the past 15 or so years to get better and better estimates of the underlying ancestral recombination graph, or the tree sequence that underlies the genetic data. And so 10 years ago, there's this huge breakthrough in the form of this piece of software called ARG-Weaver, which came from actually Adam Seipel's lab, who was on the previous slide. So maybe that was a little self-serving, that quote. I'm not saying anything about that. But this is a really cool piece of software, in part because you can draw actual posterior samples from what's called the sequential Markov coalescent, which is an approximation to the full ARG. The only issue with it is it doesn't work on huge samples. You can do like tens, dozens of samples with it. But doing 100 haplotypes or something like that would be like really pushing it. So there's been this huge effort to scale things up, and we had to make further approximations. The statistical rigor isn't there in the same way as it is for ARG-Weaver for these other pieces. But a couple of years later, there was a piece of software called Rent Plus. This one's really for hipsters. I'll talk about it a little bit more later. But then it went mainstream in 2019 with the release of these two pieces of software called Relate and TS-Infer. You can tell they're a big deal because they have their own logos. Look at these. And that scaled up ancestral recombination graph, or tree sequence inference, to the thousands or tens of thousands or even hundreds of thousands in the case of TS-Infer. And then it's continued. And just last year, we had a new one, ARG-Needle, which you can fit on biobank scale data sets, hundreds of thousands of individuals. So we've gone, in the course of just a few years, right, from being able to do this for 20 samples or so to being able to do it for 200,000. It's been a huge shift. So I'm going to make another really old reference to Heavy D and the boys. They sang, "Now that we found love, what are we going to do with it?" And my question today is, now that we found the ARG, what are we going to do with it? It's all well and good to know that, but can you actually solve some scientific questions with it? And in particular, I'm going to focus on, there's a lot of ways you could take that general question, right? I'm going to focus on how can we use it to understand complex trait variation within populations, and later in the talk also between populations and even species. So we'll talk about 3 projects. The first one by my postdoc, Vivian Link, we used estimated ARGs to essentially do haplotype mapping. and try to identify trait-associated loci in that way. Second project from my graduate students, Dan Peng and Obadiah Mulder, where we're trying to actually take GWAS information and combine it with ancestral recombination graphs to learn something about the history of complex traits. And then the third project with a research scientist, Josh Schraber, and Matt Pennell, our collaborator, we're sort of thinking about uniting existing short-term quantitative sort of statistical genetics approaches with longer timescale phylogenetics approaches. Okay, so for this first project, this is the team. I'm making so many 80s references, it's ridiculous. Vivian's positioned in here in the middle like Captain Planet. For people about my age, the rest of us were like the Planeteers. We stand around and we like summon her, and then she does useful things, and we go away. This project, what we're thinking about is an alternative essentially to genome-wide association studies. For those of you who don't know GWAS, most of you do, I'm sure, but if anyone doesn't, what you do in GWAS, right, is you run a whole lot of regressions. So you have diploid genotypes at SNPs, you have 0, 1, or 2 copies of some alternative allele, and you just regress a phenotype on that genotype value. And then you get a p-value from that regression. I'm leaving some things out, of course. You get a p-value from that regression, and you just plot all your p-values you get from all across the genome. And you get a plot that looks like this. These are the negative log 10 p-values, so that bigger is more evidence for association. And then you do further things to try and understand what's causing these peaks. And we do tons of these, right? This is several years old, but there have been many GWAS done. That's suffice to say. We learned a ton from doing GWAS. That's great, but there are some limitations to it, and these are all pretty well known. First big issue, right, is that you have to do a lot of tests during GWAS. You have to test generally hundreds of thousands of loci in a human genome. And a second issue is what's called allelic heterogeneity. So this is when you have multiple causal variants in the same locus that can sort of confuse the GWAS signal. The linkage disequilibrium between the SNP you're testing and the causal variants might be sort of diluted because you have causal variants that are in opposite LD with one tag SNP. I'll say more about that later. And then the third thing is that at least human GWAS is really built on the back of doing tons and tons of genotype imputation, which I'll talk more about later. But what that means is that you need population-specific reference panels to be able to do that, which limits the populations and the organisms that you can do this in well. So the question is whether we can use ARGs instead to identify trait-associated loci. And I'll say more what I mean by that, because that's not really a good question, because anytime you use genetic data, you're already using ARG, right? Like if I test a single variant in a GWAS, what I'm doing is testing a branch in the ARG, if you're thinking about things in that way. So is there the maybe slightly more developed question is, there's something that this representation lets us do that we couldn't do just with the genotypes. And so I'll tell you about the method we came up with. And this is not what we do, but this gives you the intuition for what we're doing. So what you could imagine is if you have a tree like this, you have these, any pair of tips here, you have a time when they coalesce, when they share a common ancestor. And you can make a matrix of all those times, right? And that's telling you something about how closely related that set of tips is, if we think about 4 haploid individuals here. We could similarly make a matrix of the differences between, if we think of haploid individuals, the differences between them on some phenotype. And if the locus we're looking at has something to do with the phenotype, we might expect that these matrices would be sort of similar. Like the closer the coalescence, the more similar on the phenotype. And it's not, again, it's not literally what we do, but that's exactly the intuition for the method I'm going to talk about. And I should point out that doing this kind of ARG-based mapping is not a new idea. It's been tried going back 20 years, and haplotype-based association methods are even older than that, which use the same general principle as what I'm going to talk about here. But some of the problems in previous attempts at this approach, well, mainly it was that you couldn't estimate the trees very well. You couldn't estimate the underlying trees. They would try to do it sort of simultaneously with the test, but you were getting samples of a few dozen individuals. And as we know now from doing lots of human GWAS, you're just not going to find many effects with samples of that size. Another big thing that happened was that as GWAS kind of gained steam, meta-analysis became a much bigger part of the picture. More and more studies proceeded by actually combining information from a lot of previous GWAS, so where the analyst might only have like summary statistics, like effect size estimates, and not the individual level genotype data. So if you wanted to fit trees, you couldn't, because you just didn't have that information from the previous samples. So I want to suggest that now is a good time to think about trying this, because for one reason is that now we have all these biobank scale data sets. If you go to ASHG, every other talk is like we have a new biobank from some other place. So there's biobanks popping up everywhere. So that means you have data sets of the size where you could actually maybe find things, and you have individual level, or at least in principle, one can get individual level genotype data from all those people. So meta-analyses are still going to be super important, but they're sort of relatively less important than they were. It's not essential to run on summary statistics. And the other reason, right, is that we've got these new pieces of software that can now estimate ARGs at scale at some level of fidelity, and so we can actually try this in a much bigger sample than we've ever been able to try before. So again, reminding you of the intuition, this is what we're basically doing. We're basically looking at whether the matrix of coalescence times is sort of similar to the matrix of phenotypic differences. I'll tell you what we really do. So what we're really going to do is we're going to, for those of you who know some statistical genetics, we're going to build an expectation of the genetic relatedness matrix, so the GRM. So if we imagine, again, a set of haploid individuals, 6 of them in this case, and I've got 3 markers from each. I have a genotype matrix that looks like this. And what I would do to compute the GRM for one locus, for one variant, is the following. I would take that vector of, this is the first one here. This vector is genotypes. I'm just going to center it first. I'm going to subtract off the mean. Now I'm going to normalize it. So what I end up dividing by is essentially what would be the standard deviation under Hardy-Weinberg equilibrium in the diploid case. But I normalize it, and then I just take the outer product. So I multiply this vector by its transpose, and I get a single variant GRM. So you can think of this as like a covariance matrix or correlation matrix for one locus. So you can see here, in this case, like C is different from A and B that are the same as each other, and that's reflected here, right? So like C is different from A and B, it has a negative number here, whereas A and B are sort of similar to each other. And when you compute a GRM from genetic variants across the genome, one way to think about what you're doing is you're just taking all these single variant GRMs and you're taking their average, you're just adding them up. But one other approach you can take, which is the approach we take here, is that you can think of the tree on which these variants hang. It might look like this. So if we knew that this is actually a pattern of shared inheritance among these 3 haplotypes, instead of weighting all of these mutations that happen to show up by 1/6 each, I can actually compute this matrix here for all 4 branches and then weight them proportionally to branch length instead. So that looks like this. So again, these matrices down here are exactly the ones that appear up here. It's just that now they're weighted by the branch length on which they appear, the length of the branch on which they appear. And what this is, we call this the EGRM, for expectation of the GRM. And it's the expectation in particular under the assumption that mutations appear on the tree as a Poisson process. Then this becomes the expectation. So if one way to think of it is if you drove up the mutation rate arbitrarily high, the GRM would converge to this expectation. So we use that matrix defined locally. So we take a local region of the genome, we figure out the EGRM for that local region. We're going to plug it into the following model. It's just like a pretty standard mixed model variance components model. So Y is our phenotype. And we're going to say that it's normally distributed, multivariate normal, with a mean determined by some fixed effects, and a variance that is partly just environmental noise, that's IID noise, but then partly structured by the local EGRM. So A here is the local EGRM. So we take like a window, like a 5 KB window of the genome, say, compute the EGRM for that window, and that's A. And then what we're interested in is sigma squared A. So it's like the variance component attributable to the shared relatedness in a specific region of the genome. And we can get that with restricted maximum likelihood. We just test whether it's equal to 0. And so doing, again, a lot of these ideas in this talk are old. Doing this with the local GRM, that is the local genetic relatedness matrix from variance, has been done for 25 years. The only difference is then we're doing it on the EGRM calculated from the trees instead. So now that I told you the idea, let's revisit what I said the issues were with GWAS and see whether we think this might have some potential to address those issues. Obviously, I picked the issues that I was going to be able to address. It's not all the issues. First issue, right, was lots of tests where if you test every one of these variants, you're actually testing very redundant information, right? These are all hanging on the same tree. They're all highly correlated with each other. So we're probably doing more tests than we need to. And if we just test trees or we just, windows, you might be able to get around that to some degree. And unsurprisingly, that's true. So this plot is showing that we do actually reduce the multiple testing burden. What I'm showing you is a bunch of simulations, and this is just these are all null simulations for like a 1 megabase region. And the y-axis is the smallest p-value of all these null tests in the 1 megabase region. And the gold line is what you get from GWAS. So, the smallest p-value ranges from, roughly 10 to the minus 4 down to 10 to the minus 2. And the blue line is our effort. And so you save basically an order of magnitude on your multiple testing burden that you have to impose by testing, 5 KB regions at a time, as opposed to testing every single variant. And that means normally in GWAS, you impose a cutoff of 5 times 10 to the minus 8. We can do something much more like 5 times 10 to the minus 7 for our p-value cutoff. Gives us a little boost in power. Second issue is allelic heterogeneity that I talked about before. This is when you have multiple causal variants in the same locus, and you can get unlucky in GWAS, and you can find something that's, for example, your text that might be in LD positively with one risk variant and negatively with another, and the signals cancel out. So that's not a rare condition at all. In fact, if you look back at the linkage era, linkage mapping is totally robust to this, and it's super common for old linkage loci found for Mendelian traits. A lot of them have allelic heterogeneity going on, and attempts to estimate this for complex traits or EQTL show that it's not rare. It's something like 20–30%, 15 to 30% maybe, with the current estimates. Although you have to take it a little bit with a grain of salt because, GWAS is not good at detecting these loci in the first place, right? So this is, if anything, a slight underestimate. So we're going to simulate some allelic heterogeneity in the following way. Vivian is going to simulate here. She already did it. She's not doing it right now. 2,000 chromosomes, simulated to have roughly a quote unquote European demographies of roughly that level of diversity, humans. And what she's going to do is in a given 5 KB window in this 1 megabase region she's simulating, 40% of the variants in that window are going to be causal for some phenotype. And that ends up being like 2 to 4 SNPs in a window. So there's like a little bit of allelic heterogeneity. We then, type 20% of the variants, that is, we get to see them and test them in GWAS and also feed them into Relate, or the ARG inference machinery. And then going to use our p-value threshold from before. So now I'll show you the results. I'm not showing you anything tree-based yet. I'm comparing 2 methods so far. So the x-axis here is essentially the effect size of the window on the phenotype, and the y-axis is the power of the method. GWAS is in gold, and what I'm showing you in pink is a local variance component method built from the actual mutations observed in that 5 kb window. So not using the tree, but doing exactly the same model. It's just we're using the GRM instead of the EGRM. And that should be robust to allelic heterogeneity, but what you can see is that we don't actually get a big boost in power compared with GWAS just from using the variance components approach. And one reason for that might be that we only have a 5 kb window, right? So we're getting a really pretty noisy estimate of the local relatedness in that region. But when you instead use the EGRM, you actually get a pretty substantial boost in power when there's allelic heterogeneity. So why is that? And our sort of theory for this is the following. Why, and not why it's better than GWAS. That's sort of, in some sense, unsurprising because our method is built specifically to be robust to allelic heterogeneity when GWAS is not. But why does using the trees beat using just the genotypes? And this is our theory for that. So say that this is my analysis window that I'm testing, like this region on some haplotypes, and there's this set of local trees describing the haplotypes in this region. Well, what this implies is if I'm doing a local GRM, I only get to use mutations that appeared in this little window. But really, there's more information in the genome available about local relatedness in this window than appears just on the genotypes that show up there, because other neighboring regions share parts of their topology with that window I'm testing. So for example, like this little tip off the end, this structure is shared, the subtree is shared between this region and the neighboring region. And so that means that any mutations that show up over here, if I know this tree structure, these mutations are actually just as informative about how long these branches are as the ones that occur in the window. And so if you know that, you can actually use the appropriate information from outside of the window you're looking at to better estimate relatedness inside the window you care about. And that's where we think the boost in power comes from. Of course, there's a question of how well any specific piece of ARG inference software captures that information. But that's the principle. I should say briefly that that was all under allelic heterogeneity. When there's only one causal variant, we don't pay a big cost. We're doing about the same as GWAS in that scenario. And I think what's going on here, we sort of do pay a cost for testing many branches at the same time that GWAS doesn't pay, but we also don't incur the multiple testing burden, and so those things kind of cancel out. This third issue is genotype imputation. So in genotype imputation, what we do is we have a sample we might care to do a GWAS in, for example. We only type a subset of the variants, and then we fill in the rest of them according to the haplotypes that we think the type things fall on. And the reason this works, right, is exactly because genetic variation is structured in this tree-like way. That's why the strong LD exists that lets you do genotype imputation very well. And in human GWAS, this is really the workhorse method. Often you're testing many times more variants that are imputed than you actually genotyped in the first place. If you think about it, this is a little bit roundabout because the reason you can do this imputation is because the ARG exists, and you sort of have to infer the ARG to do the imputation, but then rather than testing the ARG itself, you test the imputed variance. So that was kind of what got us thinking, well, what if we just tested the underlying structure? And that's sort of deeply true too, in the sense that the relate and TS-infer are actually built on the Lee and Stevens haplotype copying model that is the basis for modern imputation. So it's like literally, you're kind of inferring the ARG when you do the imputation. So I just have like an anecdote about this that has to do with this locus CREB-RF, which is a locus where there's a mutation that has a large effect size for like essentially body size measured as BMI in this case, which is not my favorite way to operationalize the phenotype. But if you do it in terms of BMI, there's this locus, the mutation that was found in Samoans that has essentially like a 1 BMI point effect per copy. So like if you're my height, that's like 8 pounds or 3.5 kilograms. So it's a pretty large effect by GWAS standards. And this mutation is really uncommon outside of Polynesia. So my colleague Charleston Chang and his group, they wrote this paper a few years ago, and they were kind of ringing an alarm bell because they said, hey, we found this mutation first in Samoans. We should be able to find it in Native Hawaiians because it's also segregating in Native Hawaiians at a lower frequency than in Samoa, but 5–10%, something like that. But you can't find it because it's not on, it's not on standard SNP panels, and it's not in high LD with anything on standard SNP panels, and it's not well imputed because we don't have population-specific reference panels until very, very recently. They didn't when they wrote this paper a couple of years ago. Don't have any population-specific reference panels for Native Hawaiians, so the imputation doesn't work well. So we thought, well, this is actually a good test case for us, right? Let's try this. And so we replicated this result. This is 5,000 Native Hawaiians from the multi-ethnic cohort, which is a cohort collected at USC, or by USC rather. So this is chromosome 5. I think I have that right. It's chromosome 5 Manhattan plot for GWAS. This is where CREB-RF is and where you should see a big peak. But again, we don't see it because none of these type SNPs are in really high LD with the actual causal SNP. You have this one little hint of one that's like, but it's still a couple of orders of magnitude away, right? If you do local GRM, you get to reduce your cutoff a little bit, but you can see we really don't get anything. And slightly irritatingly, we also don't quite get it, but I want to submit to you that we're very close. So you do see, I think, there is a peak that's way above the background for us that really isn't for the other methods. By our back of the envelope calculation, we're just under it for what our cutoff should be. Possibly that should be a little higher or a little lower, but we're right on the borderline basically at finding it. And I should say when you do this genome-wide, we find half a dozen or a dozen genome-wide significant hits, whereas GWAS doesn't find anything. And I think that's largely driven by the imputation not working as well in Native Hawaiians. So that's this section of the talk. The message, I guess, has been that if you want a better view, maybe you should climb a tree. Right. So we've known that for a couple of thousand years. These are all depictions of Zacchaeus, who shows up in the Bible in the New Testament, climbing a tree to take a look at Jesus walking by. And Jesus is very impressed by his initiative in the story. By tradition, the tree he's climbing is a sycamore fig. Although I love in this one, it looks like he's climbing a single palm frond and he's very short. So we've decided we're going to call our method sycamore. And we know what the end of our acronym is, but we're still working on the beginning. If anyone has an idea. That's the first bit. Second project, we're really going to reverse direction. And rather than trying to use the local trees to find variants associated with phenotypes, we're going to use information we already have about associations between genotypes and phenotypes and try to learn about the history of the phenotypes. So the question is, can we learn polygenic trait history? Keeping an eye on time here. And there's a bunch of ways to do this in general, right? In general, like if you have a trait that you're interested in that has a record, like an archaeological record, for example, in humans, or a paleontological record, then you can reconstruct it. Like if you care about height, standing height in humans, you can look at bones, right? And you can reconstruct. This is like within Europe, changes in estimated standing height from skeletons over, back into the Paleolithic. You can also look at ancient DNA sometimes, right? So this is estimated changes here in the lactase allele that grants sort of lactase persistence, and it's common in Europeans. This is changes within Europeans in this allele over time, calculated from ancient DNA. So that's great, but there's a lot of phenotypes we care about that don't leave a fossil record, right? And also, there's many populations and organisms we don't have ancient DNA. So what do you do in that situation? And one answer is that you can look at these coalescent trees that are encoded by the ARG, because they have information in them about population size history in particular, but also about allele frequency history, as I'll argue. So the reason for this is a coalescent tree. There's been a mutation on this one. There was a mutation from G to T and some SNP we care about. And so you can see all the T havers are inherited from that one and so forth. The shape of this tree, in particular the branch lengths of this tree, tell you something about the history of this population. And that's because the rate at which these coalescent events occur is governed by this function, little n choose 2 over big N of t. So little n here is the number of lineages that haven't coalesced, and big N is the population size at the time. So what that means is if you know the coalescent rate going back in time, then you know the population size history. That means that looking at these trees is giving you some information, right, about the population size. And I can think of that differently. I could think not just about the whole population, but also about the carriers of one allele, for example, right? If I know that this is the subtree that governs the carriers of T, then this little subtree has some information in it about the frequency of the T allele going back in time. That's the claim. So I'm not going to go into depth about, I have two broad approaches I'm going to show you for estimating allele frequency histories using these trees. Basically, one is that you can look at the tree directly and get information about the frequency of an allele among the ancestors of the sample. And if you care about the ancestors of the sample, that's great, you have your answer. The problem is that if there's natural selection, the ancestors of the sample are not representative of the ancestral population at that time. That's sort of just what natural selection is, right? If you think about it in that way. So if there's no natural selection, this principle gives us a good estimator. If there is natural selection, I can still hopefully use something about the timing of the coalescent events to learn something. So that's what we do. And we get allele frequency estimates over time, and we use them to estimate the history of population level polygenic scores. So a polygenic score is what we call them in human genetics and other fields are called breeding values or other things, estimated breeding values. A polygenic score is just a weighted sum of a bunch of genotypes weighted by their effect sizes on some trait, and you use that to predict a trait value, like in an individual. Here, I'm not going to use genotypes, I'm going to use allele frequencies because I'm trying to predict like a population average. Or I'm not really trying to predict one, but I'm thinking of it. So the betas are the allele effect sizes from GWAS, and the P's are the allele frequencies going back in time. And now here's where I layer on a zillion caveats that are really important. So we kind of might think of this very loosely as a predicted population mean trait value, but it's not that really. It only is that if there's no epistasis, no G by E, no dominance. So this is a totally, additive linear representation. We're also ignoring environmental effects on the phenotype, right? We know a lot of phenotypes change for environmental reasons over evolutionary time. Also assumes that we're tagging the causal variants consistently through time. So like either we have the causal variants or we have variants that the LD is going to stay constant with them through time, which is probably not true. And finally, one I didn't put on here, but this ignores any causal variants that have influenced the trait in the past but are no longer segregating now. So there's a whole bunch of reasons you shouldn't take this actually as a readout of the trait history. The minimal claim is that you can learn something about when selection happened. You can estimate that using the outcome of this process. Doesn't mean it was selection on the trait you care about, but you can reject hypotheses about neutrality in particular time windows. So with that, multiple layers of caveats stated, we have this population average polygenic score. This is a real simulated one. And we have these two estimators that I sort of gestured to. And without going into the details, they basically act like this. The one in green, we call the proportion of lineages estimator. That's the one that should work really well under neutrality. And we see that it kind of does under neutrality here. So sorry, this is time going into the present. There's a burst of selection in this period here. So looking back into the past, which is how you should think about these estimators, like the green window is fine until you hit the period in which selection occurred, and then it's off because the ancestors of the sample are no longer representative of the ancestral population during that. This orange one, and there's actually two that work on these principles. They're robust to selection, but they're super noisy. That's the problem with them. So that's all well and good, but this is like an interesting theory or whatever, maybe. But can we really do it? Because we don't have the real trees. So the question is, how well does this work with estimated trees? And remember, we have all these different approaches now. There's even more than this. Espalier from your colleague David Rasmussen is another one, another method that does this kind of thing. We wanted to test a bunch of these and see how well it actually works. So I'm going to show you a bunch of simulations where we have traits that are governed by 100 loci, 100 iterations each. I'm either going to show you directional or neutral evolution or a period where there's directional selection approximately 10 to 20,000 years ago, if we think of the times as being like human scale times. And I think the interesting piece is that we're going to feed every one of these software packages as many lineages as it can take, roughly, right? Where the difference between these is that, ARG-Weaver has these really satisfying statistical properties, but again, it can only take a few dozen haplotypes in. Whereas Relate and TS-Infer scale to much, much larger samples. So we should probably be expecting, if I can feed in a much bigger sample, I'll probably do a lot better with this task. This is our first set of results. So this is the simulation's neutral, and I'm using the estimator that works well under neutrality. X-axis is time. Y-axis is mean squared error. So higher is bad. Higher is a worse estimator. And the different colors are the different tree estimations. I don't have ARG needle results on this slide. We have those in the lab, but I don't have them here yet. The black is the true trees. That's about as well as you'd be able to do in principle if you really knew the actual ARG. And then everything else is kind of in a cluster. What you can see right away, ARG-Weaver does badly in the recent past, again, with only 20 tips here, and then eventually catches up and is arguably the best thing further in the past. Among the other methods, one surprising thing is that the gold arguably has a slight edge through this whole window. And that's actually Red Plus, which only takes 200 samples in, and not the 2,000 that TS-Infer would take. Okay. Under selection, this estimator breaks as we expected it to. But an interesting thing you can see, again, here's the true trees. The breakage is worse in TS infer and relate, particularly TS infer, than in the smaller samples. If I try that selection robust estimator, well, the first thing you can see is that like none of them are really up to snuff for this job when there's a selection. Right, the true trees are working okay, but with any of the estimated trees, it kind of gets thrown off during the period of selection. But once again, the relative winners here are relate and Rent Plus, the real hipster one, and ARG-weaver. The newer methods that can take larger samples don't do as well for this job. So why is this? Why don't bigger samples that are 10 times as large, why don't they help? Well, here's the reason. Remember I said before, the coalescence rate is proportional to little n choose 2, where little n is essentially the sample size, the number of lineages that haven't coalesced. What that means is if I have a huge n, huge little n, big sample, my coalescence rate is just like up in the sky. And all my tips just collapse immediately, essentially, into a smaller tree. So if I feed in a sample of 2,000 haplotypes, and a little bit into the pass, boom, that's a sample of 200 haplotypes, and it doesn't actually help us anymore. This is what an actual coalescence trees look like. And you can see here that start with a lot of haplotypes, and very quickly you're down to a much smaller sample. This figure's a little faint, pardon me. So this is true trees, and I'm just comparing sample size. So this is MSE on the y-axis, and at the timescales we're looking at, it does make a difference to only have 20 samples. But once I get to 200, I really can't tell the difference even with the true trees, right? Between 200 and 2000. The only place where it matters, I can get to zoom in and like way here next to the present, it helps to have 2000 samples. But further back, 200 does just as well. So the point here is that for this job, good branch length estimates are more important than having really big samples, if I had to pick one. And I think that raises a more general point that with any method like this that's based on estimated local trees or ARG inference, the specific things that the method does well are important. Just going for bigger or more accurate in some broad sense doesn't necessarily mean better performance for a specific job that you care about. Just for time, I'll skip this. We want to apply this across traits in the future, and we have some sort of interesting looking empirical results from the things we've done. But I want to get onto the third project, where we actually unite short and long time scale approaches. So this is all models are wrong. Some are useful. This one's both wrong and useless. This is a model of my lab meeting about 1.5 years ago. I'm there. Matt Pinnell is there. He's a phylogenetic comparative methods person. Vivian's presenting. There's other people there too, but they're not shown. There's a table. So Vivian's presenting the methods I showed you in the first part of the talk. And Matt's asking a bunch of questions about how this relates to Brownian motion and things like that, which if you're a phylogenetics person, that's immediately where you go with these things. And I think I knew just enough phylogenetics at the time to maybe like gesture at answers. But we felt like there were actually quite a lot of commonalities in the way we were coming to think about these problems. And what we came to is that, yeah, there are, as many of us know, working in evolution, there are these distinct traditions that work on different timescales to a large extent don't really hang out with each other. You might characterize one as being micro, and you would think of population geneticists and statistical geneticists and breeders maybe as being on this end, where you're interested in variation within populations, and you talk about GWAS or heritability estimation, things like that. And then there's a macro tradition where everything is about how traits change over very long timescales between species, potentially very long timescales, and the language being used is all different. You're talking about phylogenetic mixed models and independent contrasts and things like that. So given that Vivian was starting to use this tree-based approach, we started thinking about how much do these traditions actually have in common? Given this one's historically always been oriented around trees, this one not so much. So Josh Schraber, who's a really brilliant research scientist working in our labs, we just asked him, okay, just start from like totally bog standard quant gen and write down like the most general model that you can. And let's see where we get. So he does like the totally standard thing where he just, okay, the phenotype is an additive function in this model of a genetic contribution and an environmental contribution. Sure. And moreover, the genetic contribution is going to be modeled as an additive sum across a bunch of loci. Sure, whatever. And then, if you take this really seriously, it follows right away that if I want to model the covariance between two individuals, I and J, on some trait, well, it's just the covariance of their genetic components and their environmental components and so forth. Totally standard. And then if you refuse to make any further assumptions or condition on anything, you get this sort of unwieldy expression here for the covariance between their genetic effects. It's this expectation over stuff, and you have to decide at this point what's random. What are you going to call fixed? What are you going to call random? Are the effect sizes random? Are the genotypes random? Are they independent? How are we modeling like genetic similarity? Is it actually genotypes or is it some tree? You have a bunch of decisions you can make. And what Josh has shown is like starting from this kind of ugly thing, just making different choices here, you can get out, basically statistical genetics models like GWAS and GCTA models. You can get out phylogenetic linear mix models, and you can get out Vivian stuff as well, all as special cases of this same general framework. So in some sense, it's like not very surprising, right? They all kind of came from quantitative genetics in some world, so it makes sense that they all tie back. But I think it's still cool to have this connection established for a few reasons. One is that, if you know that there's this language you can talk to each other in, that means you can work better on problems that span different timescales. You can think about things like, what's the relevance of conservation at a locus for understanding, prioritization and fine mapping, which is something people want to do, but maybe don't have an explicit model for doing. Clarifies our assumptions, which is always useful. And then a kind of cute thing is that since the models are equivalent, you can just take an idea from one domain, just steal it and use it in the other domain. And it's not too hard to know when to do that, or to know how to do that at least. So we have like a little bridge, essentially. We have these two domains, and we've just built this little bridge across. So just to illustrate what I mean by that, I'm going to talk about Felsenstein's worst case briefly. So this is a super well-known paper among anyone doing phylogenetic comparative methods, where Joe Felsenstein considers a problem where you've got a hypothetical tree, species tree, where you've got like basically 20 closely related tips in each of two clades that are long separated. And if you had two trait measurements on these tips, X and Y, you might see a pattern like this. And you might conclude that with 40 observations, the apparent correlation between X and Y is strong evidence that those traits are related in some sense. But if you think about this in a different way, you don't really have 40 observations, right? You really have like 2, because like the traits changed a bunch along this lineage, they changed a bunch along that lineage, and then not so much within the tips. And so really this is like 2 independent data points. There's slightly more than that, but not much. So if you think of the 2 traits as evolving via Brownian motion, so basically like normally distributed instantaneous changes across this tree, Joe Felsenstein has a method that deals with this. But it doesn't always work. In particular, if you have like a rapid shift that doesn't obey Brownian motion along one of the branches, this won't work anymore. So Josh has simulated this. So the x-axis here is showing you how how strong of a shift has occurred at this period. And the y-axis is showing the type 1 error rate using a Brownian motion correction. And it's fine if there's a small shift, but once it gets big enough, you can get an arbitrarily high type 1 error rate from this situation. So we can use our little bridge we built and just steal a totally normal idea from GWAS and take it across the bridge of adjusting for principal components of the relatedness matrix. And once we do that, you can actually fix this thing. So with PC correction, this problem goes away, because essentially what happens is the first principal component corresponds to essentially a fixed effect on this big branch, and you get rid of the problem. So I'm not suggesting you should always throw principal components into your polygenic comparative methods, but just kind of illustrating how you can take an idea from one domain and move it over to the other, and it might be useful. Okay, so I think I'm right at time, basically. So I'll just say, hope I've convinced you that these tree representations do have useful applications, including finding QTLs, understanding trait evolution, and bridging ways that evolutionary biologists have thought about traits for a long time. And there's like a minute left. I'll take a couple questions.
You have a minute for questions. Yeah.
So you mentioned that when you building the EGR matrix, you use the branch length of the local sheets to weight original X transpose X. I'm thinking, because I haven't used any of these recommendations, ARG detection software. I was thinking what kind of result they give for the ARG reconstruction, and when you weight these matrix with the branch length, are those tips, like when they dating back to their ancestor, like are they starting at the same time point, or like, because I don't know if that's because the human genome kind of evolves quite slow, like you don't care about the sampling time of these sequences. Maybe this genome like was like sampled recently and they might experience like more mutations, so like they might have longer branch than on the local sheets.
Yeah, so to this the second question first. Yeah, everything I've talked about here is all assuming ultrametric trees. So the tip to root distance is the same for every tip. In principle, you don't have to assume that. And if you were going to incorporate ancient DNA or something like that, you have to do something different. But yeah, this is assuming all contemporary samples, but it's possible to generalize to not that case. And then to the first question, yeah, as to we're sort of still figuring out, and ARG methods keep coming out. So we're sort of figuring out with all these applications which ones work well and why, and it's kind of a moving target. Another paper I didn't talk about, and I'm a co-author on, where we use this a likelihood method for demographic inferences based on this. And whereas for the overall EGRM, relate tends to give us the best results. For that demographic inference problem, TS infer actually does better. And the reason is because for one job, what really matters is like how well you date the ancient coalescent events. And for the other, what really matters is dating the really recent ones. And they sort of do differently well in those jobs. So don't have like a unified theory of this, just to say that like there's no one across the board winner. They all sort of work well in different scenarios.
So yeah, in regards to your EGRM mapping compared to GWAS, I'm wondering if you could say something about how do you fine tune your window size or the consequences of changing that, and thinking about the weird recombination landscape in humans with, lots of hotspots and things like that.
Yeah. So, I mean, that's a good question. It's one we haven't spent a ton of time on. Like we've tried a few, like in our simulations, we try to do different sizes, and you see sort of what you'd expect in that, if your windows are bigger, then you don't pay as much of a type 1 error correction, but at some point it becomes diluted, right, and harder to pull apart from the genome-wide structure. At some point is doing a lot of work there. Obviously, I don't really have a good answer for what the right size should be, or particularly, we did everything just in physical distance. In principle, right, you might imagine doing it all in recombination distance to deal with the hotspots, but we haven't thought much about that yet.
I'm gonna let people go, but Doc's gonna be here for a little bit answering questions if anybody wants to hang out.